Welcome to the Musser Fruit Research Center, also known as the Musser Farm of Clemson University. Let me take a moment and introduce myself. My name is Jeff Hopkins. I've had the honor to be the farm manager here for the last 11 years. And today I'm going to take you on a virtual field day. We'd much rather do this in person, but given the circumstances, we'll do it electronically this time. We are a 240-acre interdisciplinary research and demonstration facility dedicated to helping the southeastern fruit industry through applied research and extension activities. Most of our research is focused on peaches. The peach team looks at the interwoven sciences of peach breeding, entomology, genetics, genomics, horticulture, pathology, physiology, sensor imaging, soils, virology, and production efficiencies. The specialists associated with these disciplines cooperate to provide research-based information to advance science and tackle challenges of the fruit industry. Peaches are an important crop for South Carolina, with 17,000 acres in production this year. The Musser Farm is the epicenter of peach research for South Carolina. We also conduct studies on apples, blackberries, and blueberries, and have various collections of interesting fruits and nuts, as well as a display of edible landscaping. We have typical facilities to enable us to complete our mission. This includes field laboratories, post-harvest storage rooms, and offices for faculty, farm staff, and students. But what really makes us unique is our people. We have over a dozen faculty from all over the world, a dedicated farm staff, and lots of students that are working on various projects all over the farm. So as you can imagine, in the middle of the summer we are a beehive of activity. Count this, collect that, measure another thing, and no, it can't wait till tomorrow is a typical summertime conversation. We are professionals, but we also have fun completing our work at the sweetest research site at Clemson University, particularly when the fruit is ripe. Today you'll be hearing from our specialists and our students about their work here at the Musser Fruit Research Center. This will give you an idea of the things that we do here. I hope that you enjoy the trip.
Good morning, my name is Guido Schnabel and I'm a plant pathologist here at Clemson University, and today I would like to tell you a little bit about our research here at the Musser Farm on Armillaria root rot management. In the southeastern United States, Armillaria root rot of peach is caused by the fungus Des Armillaria tubescens. The fungus invades the root system and colonizes eventually the below ground trunk section. The tree is deprived of water and nutrients and collapses. Early disease symptoms include poor terminal growth and undersized, curled leaves on major limbs. Infected trees may collapse suddenly after bloom and throughout summer months. Over the last decades, the tree pathogen has become the primary cause of premature peach tree decline in commercial orchards. Most rootstocks used in commercial orchards are susceptible to the disease, and management options are unfortunately very limited. In fact, chemical and biological approaches have been tried without success. But here at the Musser Fruit Research Station, we study a new cultural method that takes advantage of the inability of the fungus to grow in above-ground tree parts. Trees are planted shallow on berms to facilitate the excavation of the root collar with hose and air spade about 2 years later. Tree mortality in the root collar excavation treatment is delayed when compared to tree mortality in the grower standard treatment. The lower annual tree mortality rate results in an increase of productivity on sites with a history of Armillaria root rot problems. There are no negative effects on yield or fruit quality on trees with excavated roots. However, the modified planting system can create other horticultural challenges. For example, the berm creates uneven ground around the tree that can interfere with tree care and harvest. The furrow on either side of the berm can trap water in low areas or increase erosion due to channeling of rainwater, and the exposure of the rootstock may lead to more suckering. Current research is focused on mechanically flattening the area within tree rows to prevent erosion and to mechanically remove the soil from the root crowns. For southeastern growers, root collar excavation may be a valid option to increase orchard productivity on Armillaria root rot infested sites.
Hello, my name is Churamani Khanal and I'm Assistant Professor of Nematology in the Department of Plant and Environmental Sciences at Clemson University. My research program on peach is focused on management of nematodes associated with peach. When we talk about nematodes, the first impression that many people get is of pathogens, also known as plant parasitic nematodes. Obviously, there are a lot of plant parasitic nematodes that reduce crop yield directly or indirectly, but there are other types known as free-living nematodes that do not harm peaches. Rather, free-living nematodes play an important role in nutrient recycling, reduce disease pressure by feeding on harmful bacteria and fungus, and maintain soil health. One of my ongoing research trials here at Musser Farm involves evaluation of currently available non-fumigant nematicides against plant parasitic as well as free-living nematodes. When I say plant parasitic nematodes on peach, I'm basically talking about ring nematode. Guardian rootstock is considered tolerant to ring nematode, but under high ring nematode pressure, which is more than 50 nematodes per 100 centimeter cube of soil, peach suffer from peach tree short life, and trees usually die in 3 to 5 years. This field was planted with peaches 4 years ago, and now almost all trees are dead because of the peach tree short life. So we have this trial established where we are evaluating two different rates of nematicide, currently recommended rate and maximum allowed rate, against ring nematode and free-living nematodes. My other trial is about studying the effects of soil amendments on population dynamics of nematodes. Treatments include currently recommended rate of mulch, twice the currently recommended rate, and grower standard. For both trials, nematode populations before nematicide or mulch application are collected and compared with the nematode population in subsequent months. My future plan is to include more nematicides, including biologicals and cover crops, and see their effects on nematodes. I would like to develop a management program for a relatively new species of root knot nematode, also known as peach root knot nematode, that was first detected in South Carolina in 2019.
Hello, my name is Ksenia Gasik. I'm a peach breeder and geneticist at Clemson University. Today I'll talk to you about my peach breeding program. The Clemson University Peach Breeding Program develops high-quality peach cultivars adapted to South Carolina environment by addressing key challenges such as climate changes, consumer demands, and producer needs. Main focus is in combining disease resistance to bacterial spot and brown rot with large fruit of excellent quality. This is accomplished by searching for variability, finding new sources of desired traits, and understanding how these traits are inherited. With 15 to 20 years from cross-pollination to the release of a new cultivar, we work to predict what the industry will need in the upcoming decades. We combine conventional breeding with molecular tools to accelerate the process and improve efficiency. Traditional breeding approach in combination with marker-assisted parental and seedling selection provides efficient way of incorporating desired characters into newly developed peach varieties that address industry needs and consumer demands. Peach breeding takes long time, 15 to 20 years from the time the cross is made to releasing the new cultivar. Traditionally, hybrid seedlings have to undergo stratification before they germinate, overwinter in the greenhouse, and are planted in spring of the following year in the experimental field, where they stay for next 5 years. The first fruit is evaluated in third year after crossing for 2 consecutive years before the first level selection is made. Breeding program generates 3,000 to 4,000 seedlings per year. Field evaluations are mostly done on fruit quality, size, appearance. Those hybrids that are selected are grafted on rootstock and planted in the advanced selection block, where they are evaluated for next 5 years. Less than 2% of initial hybrid seedlings are selected and moved to the advanced selection level. Since the program's establishment in 2008, more than 400 crosses have been performed, resulting in over 30,000 hybrids planted at the Musser Fruit Research Farm. From about 600 advanced selections, 10 were clonally propagated and planted in 7 replicated trials across Southeast. After 13 years of breeding, we are now approaching an exciting time in which our own selections are being evaluated. This means in the next 5 years, peach breeding program is expected to release first varieties with high-quality fruit resistant to bacterial spot. With the release of our new cultivars, we hope the impact of our work will ensure the sustainability and profitability of the South Carolina peach industry for years to come.
Good morning, my name is Guido Schnabel and I'm a plant pathologist here at Clemson University. And today I would like to talk a little bit about our fungicide efficacy trials here at the Musser Fruit Research Center using these wonderful trees. Fungicides are an important part of our integrated approach to manage pests and diseases in South Carolina peach orchards. At the Clemson University Musser Fruit Research Center, located about 9 miles southwest of campus, we investigate the efficacy of conventional and biological fungicides against peach diseases such as brown rot and peach scab. Many of the treatments include already existing fungicides, but also some experimental products. These products are evaluated under South Carolina conditions for in-field efficacy, residual effects throughout storage, and any impact on the tree or the fruit. In the end, we look for affordable, safe, and environmentally responsible protocols to manage pre-harvest and post-harvest peach diseases. The information is used by companies to help with the registration process, by me, the extension specialist, to become familiarized with the particulars of each product, and by our producers to fine-tune spray programs. Control of pre-harvest and post-harvest fruit decay with pre-harvest sprays is determined on nectarine and peach cultivars with high susceptibility to brown rot. Trees are maintained according to grower standards, including pruning and thinning, pest control, weed control, and fertilizer application practices. However, we intentionally let disease build up during the season to make sure we have a good test. Treatments are replicated 4 times in a randomized complete block design, 1 tree per replicate. Trees are sprayed to runoff at 250 psi with a handgun at appropriate times. When fruit is at commercial shipping maturity, 100 fruit on each experimental tree are rated for the presence of brown rot symptoms and pre-harvest incidence is calculated. Additionally, 48 fruit without symptoms are picked at random and stored at 72 to 75 degrees Fahrenheit. Post-harvest brown rot is then assessed after 3 and 7 days of storage. The data is statistically analyzed, summarized, and presented to peach growers at educational meetings. Results are also used to optimize the regional pest management and culture guide.
Hello, I'm Brian Lawrence, a PhD student of Juan Carlos Melgar here at Clemson University in the Plant and Environmental Sciences Department. I study how various soil amendments, when added to peach orchards, can increase soil organic matter and improve orchard health. Current peach orchard management practices do little to increase soil organic matter, while high fertilization and irrigation rates often degrade soil structure, limiting soil biology and available nutrients. Various forms of organic matter are becoming increasingly available through waste streams from municipalities and local industries, such as poultry producers, which can supply affordable amendments to growers. Increasing organic matter within depleted soils can improve soil and tree health. And at the Musser Fruit Research Center, several projects have begun to explore orchard health after different soil amendments have been applied. All of the projects are an interdisciplinary effort led by multiple labs, including the Schnabel, Melgar, Juan, and Ye labs. We have 3 ongoing trials. The first trial is exploring the impact of food waste compost during orchard establishment. 2 rates of compost, 10 tons per acre and 20 tons per acre dry weight, were incorporated into the soil prior to making tree berms, and additional compost has been applied to the top of the berms each following year at half the original rate, and compared to control trees, which have never received compost. Additionally, we replicated this trial in 2 locations, one on a replant site and a second which had not previously been planted in peaches. The high rate of compost has yielded larger trunk size compared to the control trees at the end of the second year. Jeff Hopkins is also working with a team of professors and Musser staff on a second study. His study has added 2 rates of composted single-ground municipal mulch before and after planting a new orchard, and is currently measuring tree physiology, incidence of pathogens, including bacterial canker and Armillaria, nematode presence, and numerous soil health parameters. The mulch was incorporated into the soil to a depth of 16 inches prior to making berms, and a spreader was used to apply additional mulch on top of each row. It was easy to see differences in soil color following the addition of the treatments. A third trial is similar, but is exploring how the same composted mulch may change the soil and tree status when applied to a mature orchard. We initially added 6 inches of mulch to cover the area normally sprayed beneath the trees within each row, and have added 3 inches of mulch after the first year. As an additional treatment, poultry litter was applied at an equivalent of 3 tons per acre dry weight, then buried by a similar amount of mulch each year to supply about half the amount of nitrogen recommended annually. Within the trials, we have installed soil moisture sensors and are comparing soil water status between the amended treatments. We are also measuring changes to soil physiochemical properties, such as pH and nutrient content. Nutrient analysis of leaves, shoots, and fruits are showing which amendments can provide sufficient nutrients without synthetic fertilizer use. Perhaps most importantly for growers, we are comparing how different soil amendments impact tree growth, yield, and fruit quality, as well as their influence on tree health, with a special focus on how trees cope with bacterial diseases, namely bacterial canker and bacterial spot.
Hi, I'm Bülent Koç. I'm an associate professor in agricultural mechanization and business program at Clemson University. Here, I'll be talking about mechanical above-ground root collar excavation and removal of soil between the trees planted on raised beds.
Hi, my name is Coleman Scroggs. I'm a full-time employee here at Musser Farm, and I'm also a graduate student working with Dr. Coach.
Root collar excavation is one of the cultural management methods of Armillaria root rot. The above-ground root collar excavation involves planting trees on berms or ridges and excavating the soil around the roots after 2 years of establishment. Clearing the soil around root collar above the ground level exposes the root crown to weather conditions, including extreme temperatures and wind. This helps root crowns of the trees dry rapidly after a rain event or irrigation and reduce the water stress during a growing season. Planting trees on raised beds or berms requires tillage equipment to create ridges. While the raised beds contribute to the overall success in reducing the effects of Armillaria root rot, these raised beds are no longer required and they get in the way after the trees have matured. The raised beds between the trees in some orchards may channel water down the sides of the row. These small channels expedite the loss of soil in between the trees, increase runoff, as well as create pools of standing water after a heavy rain. These raised beds may also pose a hazard to man and equipment working near the trees after the trees are matured due to unlevel ground. We have recently developed mechanical means to flatten the soil between trees to provide a uniform and even orchard floor, which is desirable during pruning, harvesting, and other field operations.
The process of removing these berms not only gives the trees a better chance at surviving exposure to Armillaria, but also makes orchard operations more effective, safer, and easier. Applications of fertilizer and herbicides are more effective because the risk of runoff is drastically reduced. By flattening the berms, worker safety is improved. Having performed orchard operations from pruning to thinning to picking fruit on berms, I can confidently say that myself and other field workers would much more enjoy working on flat ground than having to climb up and down berms.
For this purpose, a pedal wheel was developed and mounted on a tractor-powered tillage implement. The tillage system uses hydraulic power for its operation. The hydraulic system components include a PTO pump, an oil reservoir, hydraulic lines, flow control valve, pressure gauges, and a hydraulic motor. We have also integrated an RTK GPS receiver, 2 electronic pressure transducers, and a flow meter with the system for real-time data acquisition. The measurements from the sensors and transducers are captured with a microcontroller and transmitted to a remote computer wirelessly with XP radio modules. Experiments are being conducted to determine the effects of tractor ground velocity and pedal wheel speed on torque, and energy requirements of smoothing raised beds between trees in different soil conditions. The system captures the GPS coordinates, ground speed, actual flow rate, and the RPM of the pedal wheel every second and records the data on the computer. The preliminary testing of the system components was accomplished at the Musser Fruit Research Farm. Further tests are being scheduled at the collaborating peach producers' farms. With field experiments in various soil conditions and soil types, we will be able to determine the torque, power, fuel consumption, and labor requirement of smoothing the raised beds on orchard floor. We believe these experiments will help reducing the operational cost of root collar excavation and smoothing the berms between the trees. Also, the producers will be able to assess the cost of knocking down the berms and better plan for their management operations.
Hi, my name is Juan Carlos Melgar. I'm an associate professor of Entomology here at Clemson. And today I want to spend a little bit of time discussing one of the field experiments where Dr. Guido Schnabel and I have been studying peach skin bronzing. Bronzing is a discoloration of peach fruit in the form of irregular shaped blotches. Sometimes it's just a single blotch, but some other times they can cover almost the entire fruit. It is not easy to see bronzing in the field, but under our conditions, we have seen it typically develop after rainfall and always during the very last stages of fruit ripening. In any case, symptoms intensify during storage, and that's where the problem is. Some years there are losses that exceed 50% of mid-season cultivars. Over the years and throughout different experiments, we have been trying to induce bronzing. We have seen how fruit-water relations and nutritional imbalance seem to be two factors that play a key role in inducing bronzing. While we still have not elucidated the exact mechanism, we have seen how environmental conditions inducing high fruit transpiration and orchards with high content of potassium in the soil lead to higher incidence and severity of peach bronzing. In this orchard, for instance, we have fertilized some rows with standard recommended rates of potassium and some other rows with 5 times that rate. Also, half of each row receives low irrigation, sometimes no irrigation, and the other half a high irrigation regime 5 times per week. And we are seeing how this combination causes more bronzing. Currently, one of our graduate students is looking at the nutritional imbalance that occurs specifically on those blotches on the peach skin, and it's specifically at the relation between potassium, calcium, and magnesium. Obviously, we cannot avoid rainfall, but we still need to study better the effect of certain chemistries that can reduce fruit transpiration. In any case, and while we're still working on this, we believe that management practices that follow a rational fertilization based on real orchard needs and not based on a calendar could be the first step to reduce bronzing in our orchards.
Hello, I'm Brett Blau. I'm the peach entomologist at Clemson University. Thank you for taking the time to stop by the entomology block here at the Musser Farm. Unlike everyone else at the Musser Farm, I actually want to cultivate a healthy population of insect pests to feed on my fruit. That's why they put me way over here, away from everybody else at the farm. But that's okay. Having this socially distanced orchard with plenty of peach-eating insects is really good for my research program. And let me explain why. For example, one of our native and most persistent peach pests in the southeast is the plum curculio. Plum curculio is a small, unassuming weevil that feeds on and lays its eggs in peaches. The larvae develop inside the fruit, creating an awful sight which is not easily forgotten once someone bites into an infested peach. As such, no one, neither the producers nor the consumers, wants plum curculio infested fruit. Currently, the most effective way to minimize the risk of infested fruit is to rely on frequent applications of broad-spectrum insecticides to manage the plum curculio in orchards. These recurrent insecticide applications begin soon after petal fall and last until the fruit is harvested. With the potential of spraying insecticides targeting plum curculio up to 16 times within a season, this practice is not economically or environmentally sustainable. As such, by utilizing the entomology block here at the Musser Farm, my lab is able to evaluate new, more targeted insecticides for their efficacy at managing plum curculio. We are also investigating plum curculio biology and ecology by examining their seasonal activity and dispersal throughout the season and what stimuli, such as plant volatiles, attract them to the orchards. By testing a variety of chemicals and having a better understanding of when and where plum curculio are within an orchard during the season, hopefully we can develop a more sustainable management program for producers. As such, the research in my entomology lab focuses on integrated pest management with a goal to effectively and sustainably manage insect pests in southeastern peach production. Thanks again for stopping by the entomology block. I hope you enjoy the rest of the tour.
Hi, my name is Livy Sinowitz and I'm an assistant professor of plant virology. And today I'm going to tell you about our Clemson Clean Plant Center and our Prunus Foundation. This program is associated with and supported by the National Clean Plant Network and the Peach Councils of South Carolina and Georgia. Viruses can be extremely damaging to fruit tree production, sometimes resulting in harsh eradication procedures and reduction in tree health, fruit quality, and yield. The only option to manage virus diseases is to prevent the trees from becoming infected in the first place. The best way of doing this is to ensure that trees are free from target viruses at the time of planting. This is where our Clean Plant program comes in. At the Musser farm, we maintain a foundation collection of Prunus cultivars with low chilling hour requirements suitable to the southeastern climate. In the foundation, we maintain 2 to 3 trees of each cultivar. These trees are initially tested for target viruses and also unknown viruses by using advanced molecular methods like PCR and high-throughput sequencing in order to even make it into the foundation. If viruses are detected, then the cultivar selections are subjected to virus elimination therapies using a combination of meristem tip tissue culture and heat therapy. We are currently developing these capabilities in my lab. Once determined to be clear of the target viruses, the selections are subjected to annual virus testing and are visually inspected and maintained under conditions that minimize the potential for infection, such as keeping them in a screenhouse in pots. These trees then serve as nuclear stock for the industry to propagate from. Because our indoor foundation trees cannot produce the number of buds that are needed by the tree nurseries each year, we also maintain an outdoor foundation block at the Musser farm. We also test trees on commercial farms for 3 major viruses in a long-standing program called the Southeastern Budwood Program. These programs help to reduce the prevalence of viruses in orchards and extend the life of orchards to maximize productivity and profitability for the nurseries and fruit growers. The mission of the Clemson Clean Plant Center is to continue to serve the needs of the fruit tree industry in the Southeast and nationwide.
Hi, I'm Tom Kahn, and I'm an assistant professor based at North Carolina State University in the Department of Horticultural Science. I lead an applied research program focused on serving the southeastern apple industry, which is comprised of apple growers in North Carolina, South Carolina, and Georgia. This is all developed through a multi-state agreement through Clemson University, University of Georgia, and NC State University. I'm going to introduce a trial related to some work that we're doing in apple that we've since transferred to blackberry production. In most temperate fruit crops, inadequate light distribution can have negative effects on productivity, fruit quality, and flowering. Light distribution is primarily managed in blackberry production systems by pruning and training. Inherent vigor of blackberry, particularly in the southeast United States, can result in excessive shading of the interior and basal canopy, which may have negative impacts on productivity, fruit quality, and provide a suitable environment for insect pests and pathogens. In 2021, we initiated multiple projects to evaluate the use of reflective ground covers in blackberry production systems. Reflective ground covers are woven textiles composed of polymers that are designed to be durable, and they can withstand orchard traffic and be used across multiple seasons. These ground covers reflect light from the orchard floor and enhance light distribution in the lower canopy. In addition to reflecting photosynthetically active radiation, some of these ground covers also reflect ultraviolet radiation. In other cropping systems, the deployment of reflective ground covers dramatically alters the canopy microclimate and could have many interesting or potentially useful responses in blackberry production. To our knowledge, this technology has not been formally evaluated in the southeastern United States on blackberry. To determine if this technology has merit in blackberry production, we conducted preliminary assessment of reflective ground covers in floricane and primocane production systems. In this experiment at Clemson University's Musser farm, We compared the effects of sod and reflective ground covers on canopy microclimate, bud phenology, yield, fruit quality, spotted wing Drosophila infestation, disease incidence, and vegetative growth of Osage blackberry. We're still in the process of compiling data from this study. I'd like to acknowledge the significant contributions of the multidisciplinary team of researchers that are involved in this project at both Clemson University and NC State University, along with the excellent staff at Musser Farm. We appreciate funding from NARBA to support this project.